morning, everyone, and today is the last lecture in this semester. And also, I remind you that the practice is over 2 uh, for the second group, and the last practice for the third group will be on Monday, and we conclude our course for this semester and will continue in February next year. So we also continue the last lectures topics, the regulation of growth and growth response. And as you see, the tomatoes that are green were exposed to the ethylene for 3 days on the right side. So these are ethylene exposed tomatoes, and these are normal tomatoes, and they both were picked up at the same time. And as you remember from the last lecture, the ethylene promotes the growth and ripening of the tomatoes and other fruits. And there are also additional signal molecules that affect growth and development, and biologists continue to discover new plant hormones, hormone-like signal molecules. Many of these signaling molecules are involved in defensive responses of plants to increase organisms and insects. Here we will discuss shortly the 5 groups: brassinosteroids, oligosaccharides, jasmonate, salicylic acid, and cysteine. Although steroid hormones have crucial roles in animals, we are just beginning to understand their roles in plants. The brassinosteroids are a group of steroids that function as plant hormones. Brassinosteroids are involved in several aspects of growth and development. Arabidopsis mutants that cannot synthesize brassinosteroids are dwarfed plants with reduced fertility, and researchers reverse this defect by applying brassinosteroids. Studies of these mutants suggest that the brassinosteroids are involved in multiple processes such as cell division, cell elongation, light-induced differentiation, seed germination, and vascular development. Oligosaccharins are carbohydrates that are carbohydrate fragments that consist of short branched chains of sugar molecules. They are present in extremely small quantities in cells and active at much lower concentration than hormones such as auxin. Some oligosaccharides trigger the production of photoalexins, an antimicrobial component that limits the spread of plant disease organisms such as fungi. Other oligosaccharides inhibit flowering and induce vegetative growth. So jasmonates are lipid-derived hormones that affect several plant processes such as pollen development, root growth, fruit ripening, and senescence. They are also produced uh, in presence to response the presence of insect pests and disease-causing organisms. Jasmonates may be of practical value in controlling certain insect pests without the use of chemical pesticides. For centuries, people chewed willow, that is salix bark, to treat headaches and other types of pain. Salicylic acid was first extracted in 19th century from willow bark and is chemically related to aspirin. So Hyla, do you use aspirin from headache?
What, teacher?
Do you use aspirin from headache for headache?
Yes. Yes, we use.
So the aspirin is actually a derivative of salicylic acid, and that
Mhm.
is contained in the bark of the salix or chewed willow that was used for many centuries.
Yes.
And recently, biologists have shown that salicylic acid is a signaling molecule that helps plants defend against insects, pests, and disease-causing agents such as viruses. So it looks like the salicylic acid is not only a medicine for the people, it's also medicine for the plants. But unlike the people, plants produce it themselves. So and then store it in the bark. So although many animals' hormones are polypeptides, the first plant polypeptide with hormonal properties called cysteamine was not isolated until 1991 in response to wounding by insects. Cysteamine may trigger the plant to produce molecules that disrupt insect digestion, thereby curbing leaf damage done by caterpillars and other herbivorous insects. The discovery of cysteamine in tomato leaves prompted a flurry of research that resulted in the discovery of additional polypeptides in other plants. Experiments in which different tobacco species are grafted together indicate that additional flowering uh, uh, and flower inhibiting substances may exist. Nicotiana sylvestris is a long-day tobacco plant, and a variety of Nicotiana tabacum is a day-neutral tobacco plant. So the day neutral means that the day length will not affect the flowering. Uh, when a long day tobacco is grafted to a uh, uh, short day or to the day neutral tobacco, so these are two grafted. This is a graft, that is connection between the two plants by cutting the stem slightly and uh, uh, bowing the stems. So when they are grafted together and exposed to short nights, both plants flower. The day neutral tobacco plant flowers sooner than it normally would. For many years, biologists have found evidence that a flower promoting substance that is called phlogerin may be used in the long day plant and transported to the day-neutral plant through the graft union, causing the day-neutral tobacco plant to flower sooner than expected. An intact plant may produce flowering in the leaves and transport it in the phloem to the shoot apical meristem. There, it induces a transition from vegetative to reproductive development, that is, to a meristem that produces flowers. When a botanist grafts a long-day tobacco to a day-neutral tobacco and exposes them to long nights, neither plant flowers. As long as these conditions continue, to the day-neutral plants do not flower even when they would normally do so. In this case, the long-day tobacco will produce a flower-inhibiting substance that is transported to the day-neutral tobacco through the graft union. This substance prevents the day-neutral tobacco from flowering. Although biologists have not yet isolated the chemically characterized all flower promoters and flower inhibitors, plant biologists working with Arabidopsis reported the identification of the elusive flowering signal in 2005. A gene is activated in Arabidopsis leaves and a messenger RNA molecule is transcribed from this gene. This matrix RNA molecule, thought to be phlogerine, travels in the phloem from the leaf to the shoot apical meristem. There, it is translated into protein that alters gene expression in cells at the meristem, thereby inhibiting flower development. So researchers have used Arabidopsis mutants to better understand the biology of plant hormones. For example, some mutants have defects in signal transduction, and in signal transduction, a receptor in a cell's plasma membrane converts the hormone, which is outside the cell, into a signal that causes some change inside the cell. That change ultimately leads to a physiological response. Many plant hormones bind to receptors located in the plasma membrane, triggering enzymatic reactions of some sort. Let us consider a specific example involving the plant hormone auxin. So, if we look on the how auxin works, so the step 1 is auxin binds to the auxin receptor that is located on the membrane of the cell. The binding of auxin to its receptor catalyzes the attachment of a molecule called ubiquinone to proteins known to inhibit certain genes. So this is step 2. That the proteins are attached to the genes. Whenever ubiquinone is attached to protein molecules, the cell destroys those molecules. So this is a molecule that is destroyed. So by the ubiquinone. So the next step are the genes that were inhibited by those proteins are now turned on. So proteins go to the nucleus and will turn on the genes, resulting on changes in cell development and growth. So this protein will act on the DNA that will produce another molecules that will help to growth and development. So then in the early 20th century, the two American plant physiologists, Wickman Gardner and Henry Allard, working at an agricultural research center in Maryland, US, discovered that day length, or more correctly night length, was involved in flowering. So this it was called photoperiodism. Photoperiodism is any response of a plant to the relative length of daylight and darkness. Initiation of flowering and the shoot apical meristem is one of several processes that are photoperiodic in many plants. Plants are classified into four main groups on the basis of how photoperiodism affects their transition from vegetative growth to flowering: short day, long day, intermediate day, and day neutral plants. Short day plants, also called long night plants, flower when the night length is equal to or greater than some critical period. The initiation of flowering in short day plants is due to the long, uninterrupted period of darkness rather than the short period of daylight. Uh, so the there are other factors that affect the flowering, that is the temperature, but still the light is the most important. So the initiation of flowering in short day plants so as a minimum critical night length that varies considerably from one plant species to another, but falls between 20 and 14 hours for many of the plants. Examples of short day plants are Floris chrysanthemum, cocklebur, and poinsettia, all of which typically flower in late summer or fall. Poinsettias, for example, initiate flower buds in early October in the northern hemisphere and flower about 8 to 10 weeks later, hence their traditional association with Christmas. That will start in 23 days, so on December 25. Short day plants detect the lengthening nights of the late summer or fall, and they flower at that time. Long day plants are also called short night plants, flower when the night length is equal to or less than the subcrucial length. Plants such as spinach Black-eyed Susan and Arabidopsis flower in late spring or summer and are long-day plants. These plants detect the shortening nights of spring and early summer, and they flower at that time. Intermediate day plants do not flower when day length is either too long or too short, like sugarcane and collis are intermediate day plants. These plants flower when they are exposed to days and nights of intermediate length. Some plants, called day-neutral plants, do not initiate flowering in response to seasonal changes in their period of daylight and darkness, but instead respond to some other type of stimulus, external or internal. Tomato, dandelions, spring beans, and tansy are examples of day-neutral plants. Many of these plants originated in the tropics, where day length does not vary appreciably during the year. So, in contrast, most of the short and long day plants are temperate species. So, they originally come from the temperate climate countries. The main photoreceptor for photoperiodism and many other light-initiated plant responses is Photochrome, a family of about 5 blue-green pigment proteins. A mixture of photochrome proteins is present in cells of all vascular plants examined so far. Much current knowledge of photochrome is based on various mutant Arabidopsis plants that don't produce the uh, particular type of photochrome. Biologists studying the responses of plants that do not produce an individual photochrome have concluded that the individual forms of photochrome have both unique and overlapping functions. Each member of the photochrome family exists in two forms and readily converts from one form to the other after absorption of light of specific wavelength. Once form uh, designated PR for R for red absorbing photochrome strongly absorbs the light with a relatively short wavelength. In the process, the shape of the molecule changes to the second form of photochrome that is called PFR, so designated because it absorbs far red light, which is red light with a relatively long wavelength, about 730 nanometers. When the PFR absorbs far red light, the PFR converts to the original form PR. PFR, the active form of photochrome, triggers or inhibits responses such as flowering. What does a pigment that absorbs the red light and far red light have to do with daylight and darkness? Sunlight consists of various amounts of the entire spectrum of visible light, in addition to ultraviolet and infrared radiation. Because sunlight contains more red light than far red light. However, when a plant is exposed to sunlight, the level of PFR increases. Why is photochrome important to plants? Timing of day length and darkness is the most reliable way for plants to measure the changes from one season to the next, so they don't have a calendar and they don't have a weather forecast. So I suppose that you noticed that the temperature today is rather low, about 15 degrees below zero, so that is pretty good. So I recommend you to put on some warm clothes, but the flowers don't have clothes, so they have to prepare in advance, and they don't have a forecast, they don't have internet to to look for that forecast. So but they have the photochromes. This measurement, which synchronizes the stages of plant development, is crucial for survival, particularly in environments where the climate has an annual patterns of favorable and unfavorable seasons that we have right now. So all the plants are dead or in dormancy now. So so now it's December the 2nd. So the weather outside is very shiny and there are little clouds. The temperature is 15, 17 below zero. The wind speed is about 10 meters second, and these are pretty harsh conditions for the flowering, so no flower will flower during that conditions. Uh, Photochrome is involved in the light requirement that some seeds have for germination. Seeds with the light requirement must be exposed to light containing red wavelengths. Exposed to red light converts PR to PFR and germination occurs. Many temperate species with small seeds require light for germination. Larger seeds generally do not have a light requirement. This adaptation enables the seed to germinate at the optimal time. During early spring, sunlight, including red light, penetrates the bare branches of overlying deciduous trees and reaches the soil between the trees. As spring temperatures warm, the seeds on the soil absorb red light and germinate. During the early growth, the newly germinated seedlings don't have to compete with the taller trees for sunlight. Other procedures under the influence of photochrome include sleep movements in leaves, short dormancy, leaf abscission, and pigment formation in flowers, fruits, and leaves. Zahola and Abdul, have you seen a tree near the main building of the university that still have leaves? Zahola, Jasmine, have someone seen that tree? Okay, then let's continue. Uh, so you probably have seen that light is an important mechanism of affecting seasonal responses such as the flowering. Temperature also affects seasonal responses. Certain plants have a temperature requirement that must be met if they are to flower. The promotion of flowering by exposure to low temperature is known as vernalization. The part of the plant that must be exposed to low temperature varies. For some plants, recently germinated seedlings have a cold requirement. For other plants, the moist seeds of some plants must be exposed to low temperature for a period of several weeks for flowering to occur after the seeds germinate and grow. In some plant species, the requirement of low temperature period is absolute, meaning that they will not flower unless they have been vernalized. Other species flower sooner if exposed to low temperatures, but will still flower at a later date if they are not exposed to low temperatures. Examples of plants with a low temperature requirement include annuals such as winter wheat, which grow, reproduce, and die in 1 year, and biennials such as carrots, which take 2 years to complete their life cycles. Winter wheat uh, is planted in the fall and germinates at that time. The young seedlings are exposed to cold during the winter and subsequently flower after resuming growth the following spring. Carrots and other biennials grow vegetatively the first and the first year and store surplus food in their roots. So like these are carrots, that is a biennial, and this is the growth for the first year. Unfortunately, it will not grow the second year because your mothers will take them off from the soil and chop it and put it to a salad or your plate so you can eat them. If the roots are not harvested, the plants flower and reproduce sexually during the second year. But that will not happen, believe me. Usually carrots are completely destroyed the first year. But they require the exposure to a low temperatures of winter. Carrots growing in a warm environment and not exposed to low temperatures like in Egypt continue to vegetative growth indefinitely and do not initiate sexual reproduction. So, and that is why carrots in Egypt are so large and tasty, and in temperate climate regions like in Russia, they don't have enough time to grow. So, and because of the season, they usually eat in a small size. An external stimulus to which a plant responds, such as low temperature, may be influenced by internal conditions such as hormone levels in the plant. It is possible, for example, to eliminate the low temperature requirement for flowering in biennials by treating the plant with the hormone gibberellin. So, then all the organisms, including plants, animals, and microorganisms, appear to have an internal timer or biological clock that approximates 24-hour cycle, the time that it takes for Earth to rotate around its own axis. Such internal cycles are known as circadian rhythms. Circadian rhythms help an organism to respond to the time of day, whereas photoperiodism enables an organism to detect the time of year. Why plants and other organisms have circadian rhythms. Predictable environmental changes such as the sunrise and sunset occur during the course of each 24-hour period. These predictable changes may be important to an individual organism by causing it to change its behavior or its physiological responses. It is thought that circadian rhythms help an organism synchronize repeatable daily activities so that they occur at the appropriate time each day. If, for example, an insect-pollinated flower does not open at the time of day that pollinating insects are foraging for food, reproduction will be unsuccessful. Circadian rhythms in plants affect such biological processes as gene expression, the rate of photosynthesis, and the opening and closing of stomata. Sleep movements observed in the common bean and other plants are another example of a circadian rhythm. During the day, the bean leaves are uh, horizontal, possibly for optimal light absorption, but at night the leaves float down or up, a movement that orients them perpendicular to their daytime position. The biological significance of sleep movements is unknown at this time. When constant environmental conditions are maintained, circadian rhythms repeat every 20 to 30 hours, at least for several days. In nature, the rising and setting of the sun reset the biological clock so that the cycle repeats every 24 hours. What happens if a plant's circadian clock is mutated so that the day-night cycle is not synchronized? In 2005, biologists identified mutants of the model plant Arabidopsis in which the circadian clock is not synchronized to match the external day-night cycle. The mutant plants contained less chlorophyll, fixed less carbon for photosynthesis, grew more slowly, and had less of a competitive advantage than Arabidopsis plants with a normal circadian clock. For many plants, two photoreceptors, the red light-absorbing photochrome and the blue ultraviolet A light-absorbing cryptochrome, are implicated in resetting the biological clock. Certain plants of the photochrome molecule are like clock proteins in fruit flies. Fungi, mammals, and bacteria. This molecular evidence strongly supports the circadian clock role of photochrome. The evidence for cryptochrome as a clock protein is also convincing. First discovered in plants, cryptochrome counterparts are found in the fruit fly and mouse biological clock proteins. Possibly both photoreceptors are involved in resetting the biological clock in plants. Researchers have evidence that photochrome and cryptochrome sometimes interact to regulate similar responses. The sensitive plant dramatically folds its leaves and drops in response to touch, uh, and the response, which is typically occurs in a few seconds, spreads through the plant even if only one leaflet is initially touched. When a sensitive plant leaf is touched, an electrical impulse moves down the leaf to the special cells housed in an organ at the base of the petiole called the pulvinus. The pulvinus is a somewhat swollen joint uh, that acts as a hinge. When the electrical signal reaches cells in the pulvinus, the electrical signal induces a chemical signal that increases membrane permeability to certain ions. A loss of turgor occurs in certain pulvinous cells as potassium ions exit through the now permeable plasma membrane, causing water to leave the cells by osmosis. The sudden change in turgor causes the leaves movement. Such turgor movements are temporary and reversible. The movements of potassium ions and water bulk into the pulvinous cells causes the plant pod to return to its original position. Although recovery takes several to many minutes longer than the original movement. The mechanism by which the venous flytrap loses is similar to the mechanism of the sensitive part. An electrical signal which moves more rapidly than in the sensitive part induces a chemical signal that causes a movement of potassium ions out of certain cells, followed by the exit of water. The loss of turgor causes the leaf to snap shut. So we are concluding with the lectures and uh, practical classes. That is all for this semester, and I hope you enjoyed the course and see you next year in February, except the last so today there will be no practice, so and the last practice for our third group is on Monday. Thank you for attending the lecture and see you in next year.